In this lecture, we will discuss the biosynthesis of the ABO red blood cell antigen system. I recommend accessing the free handbook on the link shown, which has very good summaries of key concepts. The ABO antigens arise due to the synthesis, or not, of oligosaccharides on the red cell membrane. These antigens are formed by addition of sugars to a precursor polysaccharide. As such, their synthesis relies on expression of genes which code for sugar transferase enzymes. As you can see from this cartoon, A and B can be expressed alone or together and are therefore codominant. Group O arises due to the lack of expression of both A or B. The genes for transferase enzymes which control their synthesis are on chromosome 9. The O allele lacks both enzymatic activities because of frame shift caused by a deletion of a guanine at position 258 in the gene, which affects a region near the N-terminus of the protein. However, two other genes, the H gene and secretor gene, are also involved in determining an individual's ABO type. This slide shows which enzyme the A, B, and H genes code for and the sugar that is transferred. The H gene is critical for formation of the A and B antigen, as 1,2-fucosyl transferase adds a fucose onto a polysaccharide precursor to form H substance. H substance is then modified by either 1,3-N-acetyl-galactosamynyl transferase, which adds a N-acetyl-galactosamine to form the A antigen, or H substance is modified by 1,3-galactosyl transferase, which adds a galactose to form the B antigen. Individuals with the O allele cannot modify the H substance and are of blood group O. As we shall see later, the H substance is still present on the red cell surface. This is a Punnett square showing the genetic inheritance pattern of the blood groups. Taking the top left as an example, it is logical that a child of parents that are both group A should be of blood group A. However, this is contingent upon the A allele being normal, i.e. not mutated, and the 1,3-N-acetyl-galactosamynyl transferase being expressed and functional. Mutations could prevent this from occurring, so would give rise to a group O blood group. Therefore, practically it is essential that good laboratory practice and standard operating procedures are followed in order to have confidence in the cross-matching data that is produced. Another complex inheritance case is available via the link on the slide. This is a very important slide, so please take time to learn this thoroughly. As a general rule for understanding which antibody is expressed by an individual, remember that if a person lacks an antigen, an antibody against that antigen will be expressed by that individual. For example, no A antigen, then anti-A antibody is present in that person's plasma. Alternatively, for a healthy person, they do not produce antibodies against their own antigens. So for individuals that express the A antigen, they do not express an anti-A antibody. The ABO system's antibodies are naturally occurring and are of the IgM type. However, they are not present in newborns until between 3 to 6 months. That forms in response to the baby putting things in its mouth, which exposes the baby to these antigens. So for individuals of blood group A, they possess functional A alleles and have the A antigen on the red cells. Therefore, they have anti-B antibodies in their serum. For individuals of blood group B, they have functional B alleles and have the B antigen on the red cells. Therefore, they produce anti-A antibodies. For individuals of a blood group AB, they possess one A allele and one B allele and have A and B antigens on the red cells. Therefore, they do not produce anti-A or anti-B antibodies. For individuals of blood group O, they do not have functional A or B alleles, and the red cells have neither A or B. They naturally produce anti-A and anti-B antibodies. Frequencies for Scotland are shown in the final column. If an individual produces an antibody against their self-antigen, i.e. a person of blood group B did produce anti-B antibodies, this would cause an immune destruction of the red cells, that is, hemolysis. Therefore, in the context of transfusion, it is ideal to give an individual their blood group that matches their own. So, for example, Group A person would be transfused with blood from another Group A person. Group AB can only receive blood from Group AB individuals. Group O can receive blood from another Group O person, as they possess antibodies that would attack red blood cells of any other group. However, Group A can receive Group O blood in an emergency situation. This is also the case for Group B individuals. Individuals of blood group O are sometimes known as universal donors, as their red cells have no A or B antigen. However, the plasma does contain anti-B and anti-A if present in high titer, has the potential to hemolyze the red cells of certain non-group O recipients, and therefore should only be used in an emergency. So we will now consider the molecular basis of AB antigen synthesis. A and B antigens are synthesized by enzymes. First, the precursor is formed, which consists of a galactose, N-acetylglucosamine, and another galactose. Next, H substance is produced by the activity of 1,2-fucosyl transferase, which adds a fucose onto the precursor. If this is not modified further, the red cells are group O, as shown in the top left-hand side. To look at this in another way, the precursor must be expressed in all red cells and made of a galactose, N-acetylglucosamine, and a galactose. The next stage is production of H substance. H gene expresses 1,2-fucosyl transferase, which transfers a fucose sugar onto the terminal galactose of the precursor. Group O only has H substance with no further modification to it or any other antigens on the cell surface. The A antigen is made by modifying the H substance. This is achieved through the expression of the A gene. The product of the A gene is 1,3-N-acetylgalactosamine transferase, which adds N-acetylgalactosamine to the terminal galactose. The B antigen is made by modifying the H substance. This is achieved through the expression of the B gene, whose product is 1,3-galactosyl transferase, which adds a galactose onto the terminal galactose of the H substance. For those who are group AB, both genes are expressed. The addition of sugars to the H substance is never 100%. Therefore, as an example, a person expressing A antigen on their red cells will also have H substance present. Thus, they have A and H antigens on the cell surface. The H gene has two variants. The dominant normal form, represented by a capital H, and the recessive mutated form, represented by a lowercase h. The enzyme 1,2-fucosyltransferase is expressed by dominant homozygotes and heterozygotes, so the precursor is modified to H substance. So in these cases, the red blood cell of all blood groups possess the H antigen, as can be seen on the left-hand side. For homozygous recessive individuals, only precursor is present on the cell surface. These individuals are called the Bombay phenotype due to where it was discovered. They will group as group O in the forward group. They will have an antibody to H, that is, an anti-H antibody. This will react with any other blood group presented. So they must have donation from another person from a Bombay blood group. This is very rare in the UK and is only stored in one bank in England. So if a person carries the gene for a blood group, that is 1,3-N-acetyl-galactosyl-amynyltransferase gene, but is also homozygous recessive for the H gene, then this person cannot form their genetic blood group, and their red cells will only express the precursor molecule. A, B, and H antigens may also be secreted into body fluids such as sweat, saliva, breast milk, and semen. Therefore, this was originally used in forensic cases. These antigens are widely distributed on a range of cells, including white blood cells, platelets, and epithelial cells. In order for the above to happen, the individuals must carry the secretor gene, thus conferring the secretor status.